Good morning. This is Sumana working as assistant professor at Department of Biochemistry, Government City College, Nayapur, Hyderabad. Today's our topic is DNA polymerase 1, 2 and 3 of E. coli. What is a DNA polymerase? DNA polymerase is nothing but an enzyme that can synthesize a new DNA strand on a DNA template is called a DNA dependent DNA polymerase or simply DNA polymerase. DNA dependent DNA polymerase. Why? Because it is calling DNA dependent means here DNA acting as the template and DNA polymerase synthesizing the DNA strand. That's why it is called DNA dependent DNA polymerase or else DNA polymerase. And both prokaryotic and eukaryotic cells contain multiple DNA polymerases. Not only 1, 2, 3, there are multiple DNA polymerases. Only some of these undertake replication called DNA replicases. Why it is called replicase means it's synthesizing the complementary strand to the template strand. That's why it is called replicase. The others are involved in subsidiary roles and are participate in repair synthesis. Not only this replicases, the other DNA polymerases participate in the subsidiary roles like repairing synthesis, or else if there is any damage it will resynthesize the DNA. And some DNA polymerases catalyze the synthesis of a DNA strand complementary to the RNA but not the DNA. Those are called RNA dependent DNA polymerase and also called reverse transcriptase. Why it is called RNA dependent means here RNA is acting as the template strand, that's why it is called, and synthesizing the DNA. So it is called RNA dependent DNA polymerase and it is called reverse transcriptase. Actually, what is the meaning of transcriptase? Transcriptase is nothing but RNA polymerase. There, RNA polymerase is also called DNA dependent RNA polymerase, where DNA acts as template and by taking DNA as template, RNA synthesis occurs. That is called transcription. And here if you see the difference, there are 3 types of enzymes: DNA dependent DNA polymerase, RNA dependent DNA polymerase and DNA dependent RNA polymerase. Wherever dependent means template and polymerase means that strand is getting polymerized. And in the E. coli, there are actually 5 types of DNA polymerases are there. DNA polymerase 1, 2, 3, 4 and 5. But here we discuss only 1, 2 and 3. In eubacteria such as E. coli, there are 3 different DNA polymerases. These are called DNA polymerase 1, 2 and 3. And what is the function of DNA polymerase 1 is that which is first discovered by the scientist Arthur Kornberg and who subsequently won the Nobel Prize for his work, that is in, and the function of DNA polymerase 1 is primarily utilizing in filling in small DNA segments during replication and also repair processes. It will just fill the gaps in the DNA during replication and the repair processes. The other enzyme DNA polymerase 2 can serve as an alternative repair polymerase. It can also replicate the DNA if the template is damaged. And DNA polymerase is the primary polymerase during normal DNA replication. So only DNA polymerase 3 acts as the polymerase enzyme, or it is called DNA replicase during normal conditions. And the other enzymes DNA pol 4 and DNA polymerase 5 are gamma family DNA polymerases and have no proofreading ability. What is the meaning of proofreading? Means whatever the nucleotides are present on the template, only the complementary nucleotides should be added during the process of replication. But because of lack of proofreading ability in polymerase 4 and 5, it can add any nucleotide complementary to the template strand and they replicate the damaged DNA. Pol 4 and Pol 5 replicates the damaged DNA. We will discuss in detail about these three enzymes. If you see in this table, the polymerase 1 have the subunit only one subunit is present in the polymerase 1, and in polymerase 2 it is unknown. The subunit number of subunits are unknown for polymerase 2, and for the polymerase 3 10 subunits are present. And here if you see the exonuclease activity, what is the meaning of exonuclease? The function of the exonuclease enzyme is it will break the phosphodiester bond between two nucleotides from one end. Okay, from one end. If it is 3' 5' exonuclease means it will break the nucleotides from the 3' directions to the 5' direction, which will break the phosphodiester bond in 3' 5' direction. And the meaning of 5' 3' exonuclease activity is these enzymes will break the nucleotides, or else it will break the phosphodiester bond between the nucleotides and removes the one by one nucleotide from the 5' end of the DNA strand, so that it will act in 5' to 3' direction. And if you consider the polymerase 1, which has both 3' 5' exonuclease activity and also 5' 3' exonuclease activity, and polymerase 3 have only 3' 5' exonuclease activity but not 5' 3', and polymerase 3 also have only 3' 5' exonuclease activity but not the 5' 3'. It means only DNA polymerase 1 has 5' 3' exonuclease activity but not for other two enzymes. And the different functions of these three enzymes is that DNA polymerase 1 will involve in DNA repair and also the gap filling, and DNA polymerase 2 will involve in the DNA repair. And polymerase 3 is the main replicating enzyme. Polymerase 3 is the main replicating enzyme. We'll discuss in detail about these three enzymes. DNA polymerase 1. The physiological function of Pol1 is mainly to repair any damage with DNA and also serve to connect the Okazaki fragments by deleting the RNA primers and replacing the strand with the DNA. Actually, what happens in the replication is there are two strands, that is 3' 5' strand which acts as a leading strand, and 5' 3' strand of the DNA acts as a lagging strand, because the replication occurs only in 5' 3' direction. So 3' 5' template can act as continuously, and 5' 3' template strand will act in discontinuous mode. So the Okazaki fragments will be synthesized, and DNA polymerase can't directly add the nucleotides to the template strand. It requires free 3-hydroxyl group which is provided by the RNA primer. So in the leading strand only one primer will be there, but when it comes to the Okazaki fragments, there for each Okazaki fragment there is a RNA primer. So all those RNA primers should be removed and those should be filled with the DNA. That function will be done by the DNA polymerase 1, and it has the 5' 3' exonuclease activity of Pol 1 is independent of both 3' 5' exonuclease and its polymerase activities. And when you use the enzymes, that is proteases, example subtilisin and a trypsin, which cleaves the polymerase 1 into two fragments, one large subfragment and the other is smaller fragment. A large fragment is at the C terminal, that is at the carboxyl end of the protein chain. A larger C terminal or clenofragment, which is also called clenofragment, which contains both the polymerase and also the 3' 5' exonuclease activity. It has the polymerase and 3' 5' exonuclease activity. And when it comes to the smaller fragment of the DNA polymerase, which is N-terminal fragment, contains 5' 3' exonuclease activity. It has only one function, that is 5' 3' exonuclease activity. That's why in the previously we discussed that the 5' 3' exonuclease activity of Pol 1 is independent of both 3' 5' exonuclease and polymerase activities. And DNA polymerase 2. This DNA polymerase 2 is encoded by the gene Pol B. And which involves with the DNA replication in some capacity, synthesizing chains of nucleic acids. And this polymerase 2 can serve as an alternative repair polymerase. What is the main repair enzyme? Polymerase 1. And this polymerase 2 can act as alternative repair polymerase, and it can also replicate the DNA if the template is damaged. Next enzyme is DNA polymerase 3. And this DNA polymerase 3 holoenzyme, which is a complete enzyme, is the major replicative polymerase for both leading and lagging strand synthesis. And this Pol 3 holoenzyme is a huge multi-protein complex which has 10 distinct polypeptides. Those are alpha, epsilon, theta, tau, gamma, delta, chi, psi, beta and delta epsilon. All these 10 subunits are together during the replication process. If you see in the diagram, here it is a dimeric component which has two complexes, and here alpha subunit is there, beta, epsilon, theta, tau, and this complex is for lagging strand and this is for leading strand synthesis, and these are together with the tau subunit. And the different functions of different subunits are here. We will discuss the alpha subunit, which is encoded by the gene DNA-E, has the polymerase activity. Mainly alpha subunit has the polymerase activity. Next, epsilon subunit, which is coded by the gene DNA-Q, has the 3' 5' exonuclease activity. 3' 5' exonuclease activity. And theta subunit, which is coded by the gene whole E, stimulates the epsilon subunits proofreading. Next, beta subunit, which is coded by the gene DNA-N, acts as a sliding DNA clamps. They keep the polymerase bound to the DNA. Always the enzyme should be with the template strand. So this will be done by the subunit beta. And tau units are coded by the DNA X genes, which acts to dimerize the two of the core enzymes. Each core enzyme consisting of alpha, epsilon and theta subunits. Next, gamma unit, which is coded by the gene DNA X, which acts as a clamp loader for the lagging strand, not for the leading. Here it will acts as a clamp loader for lagging strand. The delta is involved in copying of the lagging strand, and chi, which is coded by the gene HOLC, and psi, which is coded by the holo C, which form a 1:1 complex and binds to tau or gamma. Chi also mediate the switch from the RNA primer to DNA. And DNA polymerase 3 has very high catalytic potency and the processivity. What is the meaning of processivity? Is that processivity refers to the number of nucleotide additions per binding event between the DNA polymerase and the template. Once the enzyme bind with the DNA template, how many number of nucleotide added per each binding is called processivity. And there are two types of enzymes, non-processive DNA polymerase and processive DNA polymerases. And these non-processive DNA polymerases incorporate one or few base pairs per DNA binding event. Only one or few nucleotide additions per binding. And processive DNA polymerases incorporate thousands of nucleotides per DNA binding event. Per each binding, it can incorporate thousands of nucleotides per binding event. And this polymerase 3 enzyme can extend the DNA chain with a high processivity. That is, per each binding, more than 500 kb of DNA can be continuously synthesized without dissociation of the polymerase from the template. More than 500 kb of kilo base pairs of DNA can be continuously synthesized without dissociation of polymerase from the template. And it has high catalytic efficiency. What is the meaning of catalytic efficiency? Is that the velocity of chain elongation is about 1000 nucleotides per second at 37 degrees centigrade. Per each second, it can add 1000 nucleotides. That's why it has high catalytic efficiency. And here if you see here, the replication complex is an asymmetric dimer. Why it is called asymmetric is because for the leading strand it has only 5 subunits, and for the lagging strand it has, this is 5 complex and this is 5. Here two complexes are together. That's why if DNA polymerase 3 forms the asymmetric complex, and this is for the leading strand, this complex is for lagging strand. These two are together with the two subunits which maintains the dimeric structure of the DNA polymerase 3. And the catalytic core means which is the main function of DNA polymerase 3, composed of alpha, epsilon and theta subunits. Only the catalytic core is with the alpha, epsilon and theta. This is the core subunit, contains the polymerase activity and also the 3' 5' exonuclease activity for proofreading. Proofreading is nothing but addition of the correct nucleotides complementary to the template strand. And a dimer of beta subunit. Here you can see beta subunits are in the form of dimers. It means two beta subunits forms a ring shaped structure. Here you can see this ring. The dimer of beta subunit forms a ring shaped structure and acts as a sliding clamp during replication process. Only with the help of beta subunit, the enzyme can slide on the template strand, and loading of the beta clamp onto the DNA is facilitated by the 5 subunit subassembly called gamma complex. This is the gamma complex, which consisting of gamma, delta, chi, psi and delta epsilon. And the dimerization component, that is tau, which causes the two core polymerase molecules at the fork to link together and form the dimer. Only these two subunits are useful for the maintenance of the dimeric structure of the polymerase 3 enzyme. Here if you see in the table, the different functions are explained here. The comparison of three enzymes of DNA polymerases, that is 1, 2 and 3, here you can see. Here mistake is there, it is 3 actually, DNA polymerase 3. And the polymerization activity. Always the polymerization occurs in 5' to 3' direction. It means it will add the nucleotides in 5' to 3' direction. This function is for all the three enzymes of DNA polymerase. The exonuclease activity, this is 3' to 5' exonuclease activity, and the 3', 5' exonuclease activity is for all the three enzymes. It means it removes the nucleotides by breaking the phosphodiester bond from 3' end to 5' direction. And the exonuclease activity of 5', 3', which has only for DNA polymerase 1 but not for polymerase 2 and polymerase 3. Because of this activity only it can remove the RNA primers and can add the DNA gap fillings during the replication process, which is done by DNA polymerase 1. And use of primer single strands. It means only DNA polymerase can act primer single strands. During replication it can remove the primers. And use of nicked duplex or helix. Nicked duplex or helix means wherever the gaps are there, those are filled by the DNA polymerase 1. And the molecular weight of the DNA polymerase 1 is 1,09,000, which is a single chain. It means it consisting of only single polypeptide chain or single subunit. And the molecular weight of the polymerase 2 is around 1,20,000, and which is also a single chain. And the polymerase 3 consisting of molecular weight of more than 2,50,000, which is a heteromultimeric chain which consisting of around 10 subunits. And the number of molecules of this DNA polymerase per cell are, for polymerase 1 it is around 400 molecules, and polymerase 2 is now unknown, and for the polymerase 3 the number of molecules per cell are 10 to 20. And each of these enzymes are coded by the genes. That enzyme DNA polymerase 1 is coded by the gene Pol A, and polymerase 2 coded by the gene Pol B, and polymerase 3 enzyme is coded by the gene, this polymerase 3 enzyme is coded by the gene Pol C, and with the each subunit is coded by a different gene like DNA E, DNA N, DNA ZX, DNA Q and DNA T. And number of nucleotides polymerized at 37 degrees centigrade, that is molecules per minute, will be added by the DNA polymerase 1 are up to 1000 nucleotides, and for DNA polymerase 2 it is only up to 50 nucleotides can be added per minute. And which DNA polymerase 3, which is a main replicating enzyme, can add up to 15,000 molecules per minute around at 37 degrees centigrade. And the affinity for nucleoside triphosphate precursors, which has low affinity for both DNA polymerase 1 and 2, and for the polymerase 3 has high affinity for nucleoside triphosphate precursors. In this way, these three enzymes will be involved in the replication and repair process of the DNA. Only polymerase 3 is the main replicating enzyme, Polymerase 1 to are for repair, and polymerase 1 is mainly removes the RNA primers and fills the gap during replication of the Okazaki fragments. And thank you to all.